This lecture focuses on the core microbiology concepts required for GAT-B, Unit 2, Part B, which is one of the most important and scoring section in the exam. The topics included here are foundational in nature, meaning that once you understand these clearly, you can solve both direct and application-based questions. Microbiology as a subject connects multiple disciplines such as biotechnology, medicine, agriculture, and environmental science, making it essential not only for the exam but also for higher studies. In this session, we will systematically cover all the topics starting from viruses and microbial diversity of growth, metabolism, and diseases. The goal is not just to memorize facts, but to understand the logic behind each concept. This approach is particularly important for GAT-B, where questions often test conceptual clarity rather than rote learning. By the end of the session, you should be able to interlink topics and apply your knowledge efficiently. Preparation for GAT-B requires a structured and concept-driven approach rather than random studying. The key idea is to focus on building strong foundations first, followed by consistent practice of MCQs and previous year questions. Many students tend to memorize definitions, but the exam often asks questions in an applied manner. So understand the reasoning behind concepts become crucial. A planned preparational strategy helps in covering the syllabus efficiently and avoids last minute confusion. In addition to conceptual clarity, regular revision and testing are equally important. Mock tests help identify weak areas and improve time management skills. Proper guidance throughout the preparation process, from registration to counseling, ensures the students stay on the track. Resources such as clear notes, doubt solving supports, and updates exam tips play a significant role in the improving performance and confidence. This unit includes several interconnected topics that together forms the foundation of microbiology. These include viruses, their structure and classification, microbial diversity, bacteria, algae and fungi, methods used in microbiology, microbial growth and nutrition, respiration processes, nitrogen fixation, and microbial diseases, along with host-pathogen interactions. Each of these topics contributes to understand how microorganisms function and interact with their environment. This is important to note that these topics are not just isolated, rather they are interconnected. For example, microbial growth depends on nutrition, while respiration is directly related to energy metabolism. Similarly, nitrogen fixation is carried out by specific microorganisms linked to the microbial diversity. Understand these connections will help you answer integrated and higher level questions. Introduction to microbiology. Microbiology is the scientific study of microorganisms which include bacteria, virus, fungi, algae, and protozoa. These organisms are microscopic in size but have a massive impact on living systems and the environment. They play essential role in the nutrient cycle, decomposition, and maintaining ecological balance. In addition, microorganisms are widely used in industries for the production of antibiotics, enzymes, and fermented products. Microorganisms can be broadly classified into prokaryotic organisms like bacteria, eukaryotic organisms like fungi and algae, acellular entities like viruses. This classification is fundamental and frequently tested in exams. Understanding the structure and function of these organisms help in controlling diseases, improving agriculture productivity, and develop biotechnological application. Thus, microbiology is the highest applied and relevant field of study. Viruses. General characteristics. Viruses are unique biological entities that differ significantly from cellular organisms. They are described as obligate intracellular parasites because they can only replicate inside a living host cell. Outside the host, they remain inactive and do not exhibit any characteristics of life. This dependence on host machinery makes them fundamentally different from bacteria and other microorganisms. Viruses possess either DNA or RNA as the genetic material, but never both. They lack essential cellular components such as ribosomes and metabolic pathways, which mean they cannot generate energy or synthesize proteins on their own. Due to these characteristics, viruses are often considered to lie at the boundary between living and non-living organisms. Their simplicity and dependency make them highly efficient infectious agents. Structure of viruses. The structure of virus is relatively simple but highly specialized for infection. At the core lies the nucleic acid, which can be either DNA or RNA and may be single-stranded or double-stranded. This genetic material carries the information required for replication and infection. Surrounding the nucleic acid is a protein coat known as the capsid, which protects the genetic material and helps in delivering it into the host cell. Some viruses also possess an outer lipid envelope derived from the host cell membrane. This envelope contains glycoproteins that play a crucial role in recognition and attaching to the host cell. Additionally, certain viruses have accessory structures such as tail fibers in bacteriophages or enzymes like reverse transcriptase in retrovirus. These structural features enhance the virus's ability to infect and replicate within host cells. Classification of viruses. Viruses are classified based on multiple criteria, with the most important being the type of genetic material they contain. DNA viruses and RNA viruses differ in the replication mechanism, which is a key concept in virology. In addition to genetic classification, viruses are also categorized based on their morphology and structural features. They may have helical or complex shapes. Another important distinction is between envelope and non-envelope viruses. Envelope viruses are generally more sensitive to environmental conditions, while non-envelope viruses are more resistant. These classifications are crucial for understanding virus behavior and pathogenicity. Microorganisms are classified using various criteria such as morphology, metabolic characteristics, and genetic composition. This classification helps in identifying and studying different types of microorganisms systematically. It also provides insights into their ecological roles and applications in different fields. The major groups of microorganisms include bacteria, algae, and fungi. Bacteria are prokaryotic and structurally simple, while algae and fungi are eukaryotic and more complex. Each group has unique features that distinguish it from others. Understanding these differences is important for topics such as microbial diversity, metabolism, and disease causations. Bacterial diversity. Bacteria exhibit a wide range of diversity in terms of shape, metabolism, and environmental adaptation. Based on shape, they can be classified as cocci, spherical, bacilli, rod shape, and spirilla, spiral shape. This morphological classification is one of the simplest way to identify bacteria and is often used in preliminary analysis. Bacteria can also be classified based on gram staining into gram-positive and gram-negative types, which differ in the cell wall structure. Additionally, they are categorized based on oxygen requirements into aerobic and anaerobic bacteria, and based on nutritional modes into autotrophs and heterotrophs. Some bacteria can even survive in extreme conditions, making them important in ecological and industrial processes. Algal diversity. Algae are photosynthetic eukaryotic organisms that are primarily found in aquatic environments. They contain chlorophyll and are capable of performing photosynthesis, thereby producing oxygen and serving as primary producers in aquatic ecosystems. This means they are a crucial component of the food chain and global oxygen cycle. Algae can be unicellular or multicellular and are classified into different groups such as green algae, brown algae, and red algae based on their pigmentation and other characteristics. Each group has distinct features and ecological roles. Their ability to convert sunlight into chemical energy make them essential for sustaining life in aquatic habitats and maintaining ecological balance. Fungal diversity. Fungi are eukaryotic, heterotrophic organisms that obtain their nutrition by observing organic matter from their surroundings. Unlike plants, they do not perform photosynthesis and instead depend on decaying organic material or living host for nutrition. A key structural feature of fungi is their cell wall, which is composed of chitin rather than cellulose. This distinguishes them clearly from plants and is an important point of exam. Fungi exist in different forms such as yeast, unicellular, molds, filamentous, and mushrooms, multicellular. They reproduce through both asexual and sexual methods, producing various types of spores. Fungi play a crucial ecological role as decomposers, breaking down dead organic matter and recycling nutrients. Additionally, they are widely used in biotechnology, particularly in the production of antibiotics like penicillin. Methods in microbiology. Microbiology relies heavily on laboratory techniques to observe, identify, and study microorganisms. One of the primary tools in microscopy, which includes light microscopy for basic observation and electron microscopy for detailed structure analysis. These tools allow scientists to visualize microorganisms that are otherwise invisible to the naked eye. Another important aspect is sterilization, which ensures that unwanted microorganisms are removed or destroyed. Methods such as autoclaving use high pressure steam to achieve sterilization, when filtration is used for heat sensitive materials. Additionally, culture techniques using agar plates or liquid media are used to grow microorganisms under controlled conditions. Staining techniques like gram staining and acid fast staining are essential differentiating between types of bacteria. Pure culture techniques. In natural environments, microorganisms are usually present in mixed populations, making it difficult to study a single species. Pure culture techniques are used to isolate and grow a single type of microorganism so that its characteristics can be studied in detail. This is essential for identification, research, and industrial applications. Common methods for obtaining pure cultures include the streak plate, spread plate, and pour plate techniques. These methods help in separating individual cells, which then grow into distinct colonies. Each colony originates from a single cell, ensuring genetic uniformity. Mastering these techniques is important for microbiological research and is also frequently tested in competitive exams. Microbial growth refers to an increase in the number of cells rather than increase in size. When microorganisms are grown in a controlled environment, they exhibit a characteristic growth curve consisting of four distinct phases. These phases help in understanding how microorganisms respond to environmental conditions over time. The lag phase is the initial stage, where cells adapt to the new environment without dividing. This is followed by the log or exponential phase, where rapid cell division occurs. The stationary phase occurs when the rate of cell division equals the rate of cell death, usually due to limited nutrition. Finally, in the death phase, the number of dying cells exceeds the number of new cells formed. Understanding this growth pattern is essential for both theoretical and practical applications. Microbial growth is influenced by several environmental factors that determine the rate and extent of growth. Temperature is one of the most critical factors, with different microorganisms adapt to different temperature ranges. Each organism has an optimum temperature range where growth is maximum. Other important factors include pH, oxygen availability, nutrient supply, and water activity. These factors collectively determine the survival and growth efficiency of microorganisms in different environments. Microbial nutrition. Microorganisms require nutrition for energy production, growth, and cellular functions. Based on their carbon source, they are classified into autotrophs, which use carbon dioxide, and heterotrophs, which rely on organic compounds. This is classified helps understanding how different microorganisms obtain their basic building materials. In addition to carbon, microorganisms also differ in their energy source. Phototrophs obtain energy from light, while chemotrophs derive energy from chemical compounds. These nutritional classifications are important for understanding microbial metabolism and ecological roles. They also help in identifying microorganisms and predicting their behavior in different environments. Aerobic respiration is a metabolic process in which oxygen acts as the final electron acceptor. It involves a series of biochemical reactions that convert glucose into energy in the form of ATP. The process includes glycolysis, the Krebs cycle, and the electron transport chain, each contributing to energy production. This type of respiration is highly efficient and produces a large amount of ATP, typically around 36 to 38 molecules per glucose molecule. The end products of aerobic respiration are carbon dioxide and water. Because of its efficiency, aerobic respiration is preferred energy generating process in organisms that have access to oxygen. Anaerobic respiration occurred in the absence of oxygen and is less efficient than aerobic respiration. In this process, organisms use alternative electron acceptors such as nitrate or sulfate instead of oxygen. This allows them to generate energy even in oxygen deficient environment. Another form of anaerobic metabolism is fermentation, where non-electron transport chain is involved. Fermentation produces less ATP, but also cells to survive in anaerobic conditions. Common examples include lactic acid fermentation and alcoholic fermentation. Understanding these processes is important for both microbiology and industrial applications like food production. Nitrogen fixation is a crucial biological process in which atmospheric nitrogen is converted into ammonia, a form that can be utilized by plants. Since atmospheric nitrogen is not directly usable by most organisms, this process plays a vital role in maintaining the nitrogen cycle and supporting life on Earth. Nitrogen fixation is carried out by specific microorganisms, including free-living bacteria like Acetobacter and symbiotic bacteria like Rhizobium, which forms association with plant roots. The process is catalyzed by the enzyme nitrogenase, which is highly sensitive to oxygen. This makes nitrogen fixation a complex and tightly regulated process essential for soil fertility and agriculture productivity. Microbial diseases. Microorganisms can act as pathogens and cause disease in humans, animals, and plants. These diseases can be caused by bacteria, viruses, or fungi, each having different mechanisms of infection. For example, bacterial diseases include tuberculosis, viral diseases like influenza, and fungal infections like candidiasis. The ability of a microorganism to cause disease depends on virulence factors, which enhance its capacity to invade host tissues and evade the immune system. Understanding microbial diseases is important not only for diagnostics and treatment but also for prevention and control strategies. This topic is highly relevant for GAT-B as it combines concepts from microbiology and immunology. Host-pathogen interaction refers to the dynamic relationship between a microorganism pathogen and the host organism it infects. The outcome of this interaction determines whether a disease will occur or not. When a pathogen enters the host, it attempts to establish infection by adhering to host tissues, invading cells, and multiplying. At the same time, the host activates defense mechanism to eliminate the pathogen and prevent damage. The host defense system consists of innate immunity, which provides immediate but non-specific protection, and adaptive immunity, which is specific and develops over time. The balance between the strength of the pathogen and the effectiveness of the host immune response determines the severity of the infection. If the pathogen overcomes the host defenses, diseases develops; otherwise, the infection is controlled or eliminated. Virulence factors are specific characteristics or tools that enable a pathogen to infect a host, cause disease, and evade the immune system. These include structures like adhesions, which help microorganisms attach to the host cells, and capsules, which protect them from being destroyed by immune cells. Without these factors, many pathogens would not be able to establish infections effectively. In addition to structured components, pathogens also produce toxins and enzymes that damage host tissues and facilitate the spread of infection. For example, toxins can disrupt normal cellular functions, while enzymes can break down host barriers and allow deeper invasion. The presence and strength of these virulence factors directly influence the pathogenicity of a microorganism, making them an important concept in understanding infectious diseases. Summary: this unit provides a comprehensive understanding of microbiology, starting from the basic structure and classification of viruses to the diversity of microorganisms such as bacteria, algae, and fungi. It also covers essential laboratory techniques used to study microbes, along with the factors affecting microbial growth and nutrition. These foundational concepts are critical for understanding how microorganisms live and function. Additional to the unit explains important metabolic processes such as aerobic and anaerobic respiration, as well as nitrogen fixation, which plays a key role in ecological balance. Finally, it addresses microbial diseases and the interaction between host and pathogen, highlighting the importance of immune responses and virulence factors. Together, these topics form a complete framework for understanding microbiology in the context of GAT-B. Exam tips: GAT-B focus. For effective preparation in GAT-B, it is important to focus on higher yield areas such as classification systems, including gram staining and the classification of viruses. Understanding the microbial growth curve and being able to interpret it also essential, as questions are often asked in a conceptual manner rather than directly. Similarly, differences between aerobic and anaerobic respiration should be clearly understood. Students should also pay Special attention to nitrogen fixation, particularly the role of the enzyme nitrogenase, as well as host-pathogen interactions and virulence factors. Practicing previous year questions and mock tests is crucial for identifying important patterns and improving problem-solving speed. A combination of concept clarity, regular revision, and practice is the key to score well in this session. Consistent preparation and regular revision are the most important factors for success in competitive exams like GAT-B. Instead of studying everything at once, it is better to follow a structured plan and revise topics multiple times. Strong concepts built over time will help in retaining information and applying it effectively during the exam. Staying updated with preparation strategy, solving practice questions, and clearing doubts regularly can significantly improve performance. A focused approach with clear understanding rather than rote memorization will lead to better results. With proper effort and strategy, this unit can become one of the most scoring sessions in your exam.